Getting towards the end now of our Cafe Scientifique presentations. I'm Graham Schimmel, Executive Director at Bigelow Lab. Welcome this evening. Before I introduce this evening's speaker, just a few announcements. The first is this green piece of paper that's on the seat that many of you are sitting on. We're very interested in trying to improve the Cafe Scientifiques and to think about how we can structure these for next year. So we really welcome your feedback for this. It's fairly self-evident what you have to do. If you're not looking at the correct page, there's a list of the talks that you've heard, and number 3 on this list is the one to vote for. Seriously, we do welcome your feedback on this. There's an envelope if you want to post it, or just put it in the envelope and leave it on the seat. We can pick it up. It's also going to be on the Bigelow website as of next week, so if those of you who like to do these things online, please feel free to do that as well. We do really value your feedback. For some of you who may be new, there's a visitor book on the table to my right at the back. We'd be very pleased to have your name and address so that we can keep in touch with you. So thank you very much. So this evening, it's a great pleasure to introduce you to Dash Masland. We've got to know Dash at Bigelow Lab as one of the key research staff members of the Single Cell Genomics Center, working with Dr. Romina Stepanauskas, who's here this evening. Dash joined us with a BA in biology from Colorado College, and she's been studying gray seals off the coast of Maine as part of her master's program in marine biology down in Biddeford at the University of New England. As well as undertaking the laboratory studies, she's a certified dive master and research scuba diver, and has multiple sailing certificates as well. Dash, I think, epitomizes one of the people that's always been interested in marine biology from a young age. I think, if you want a role model to follow and how to get involved in the science and try to understand how to bring it to life for young people, Dash is the kind of person who's a great example for that. And she really has epitomized that by applying for and winning the support of the National Geographic Foundation for those studies that she's going to talk to us about this evening, which are already underway. So, great expectations this evening. No pressure at all. No pressure. I told her already you're great fun. You're a very friendly audience, and you'll ask all sorts of hard questions at the end. But over to you.
Me, and I'm really excited to be here. And I know Graham wants the presentation pre-checked, but I don't know, what number is this week?
You can get a high score.
I think with the survey tonight was pretty good. Okay, so the title of my presentation is Expedition Granted, National Geographic DNA Dietary Studies in the Endangered Hawaiian Monk Seal. So I'm just going to kind of take you guys through my relationship with National Geographic and how I got involved with them, and then talk a little bit about the Hawaiian monk seal and the project I'm working on with that species. So, the story starts here. This is a harbor seal. And one of the things I get a lot when I talk about seals, in fact I do seal research, is how cute they are. And so you can see the Hawaiian monk seal is also really, really cute, and the harbor seal is really, really cute, and I started studying the great seal. I don't tend to get that same reaction with the great seal, but I think that it's adorable, and it starts its life out quite adorable as well. So fair enough, it does have some cute stages of life. But my story with the gray seal starts with graduate school. I enrolled at UNE, and I kind of went in right out of college, ready to go, do some research, super excited, and I get to my advisor's office on the first day of grad school, and I sit down, and I look at her, and she says, well, Dash, what do you want to study? And I was like, what do you mean, what do I want to study? I mean, I thought we kind of, worked on that together, and I don't know if those of you familiar with master's programs in the science sciences, you tend to kind of maybe hop onto your advisor's project or a PhD's project, and you have a component of it and you make it your own, and it's definitely very real and good science, but there's usually kind of backbone, and I didn't have any of that. So I thought to myself, okay, well, here we go. So I got to go out, and I just started. She's a pinniped biologist in Maine, so I went out and I just started reading everything I could about pinnipeds in Maine. And there really are the only species we saw here in Maine year-round: the gray seal and the harbor seal. So I started learning about gray seals, and there's 3 distinct populations. There's in the western North Atlantic, the eastern, and the Baltic Sea. And what's really interesting about gray seals is that in the early 20th century in New England, there was a bounty put in place for their removal because it was thought that they were a nuisance with fisheries. So basically what happened was people got paid to kill them, and we extirpated them completely from the US. So in about the early 20th century, there was no longer gray seals here. That bounty was finally lifted in the 60s, and the Marine Mammal Protection Act was put in place in 1972. So this really helped protect all seals in Maine, New England, the US. But at the time, there still weren't a lot of gray seals. And what ended up happening is in Canada, there was a fairly healthy population on an island called Sable Island. I don't know if any of you are familiar with Sable Island, but it's a huge gray seal hotspot. There's thousands of them. And that population got really, really large, and the gray seals kind of started coming back down here. And because there was no longer any hunting pressure on them, they were able to repopulate New England's waters. So the last survey we did was in 01. Well, I wasn't a part of it, but the last survey that was done on population was in 01. And there was about 1,700 gray seals in Maine at that time. And then there were very, very, very small breeding populations here, just a couple hundred. And then south of Maine, so this is kind of like New Hampshire down to Long Island is their range currently. And quite a while ago, there was about 6,000 and about 1,000 pups. So this is not a huge population of seals, but it is definitely up and coming. And that's really cool for a biologist because this population was removed and completely gone from our ecosystem, and then it's really interesting to see how it comes back and affects the ecosystem and how things might have shifted. So the diet of these seals is widely studied throughout their range. There's tons of information from Canada and from Europe on what these gray seals are eating, but at the time there was no research on the diet of gray seals in Maine. So that's what I decided to study. And I'm all excited, and I get all my papers together and all my notes, and I go back in, I sit before my advisor and say, okay, this is what I'm going to do. I'm going to study diet of gray seals in Maine. We have all the methodology worked out, and she looks at me and she goes, great, Dash, how are you going to fund this? And I was like, oh, okay, money, right, that's important for research too. So I basically spent the entire first year of graduate school looking for funds. And I stumbled across a program through National Geographic called the Young Explorers Grant. And what this is, it's a program for not necessarily students, but for young adults ages 18 to 25. And they fund a couple different areas. There's research, conservation, and exploration. So this fellow here was documenting migrations. They funded a fellow who went across the whole US filming festivals. So basically any young explorers that have a project idea that would contribute to the body of knowledge, they consider funding. So I applied and I received a Young Explorers Grant, which was really exciting, and it kind of started my relationship with National Geographic. I also want to recognize the Darling Marine Center because I also received a visiting graduate student award through them, and I'm incredibly grateful for that. And it's also a very neat program. They give, instead of giving funds to students, they actually give credit to use at their facility. So I got to use credit for their boats to help get me out to the islands to access the gray seals. So that was a huge component of my research funding. And then the UNE did, in fact, have a little bit of money that I could access as well. So then I finished grad school, which was great. 3 years instead of 2, that was okay. And I ended up here at Bigelow working with Ramunis in the Single Cell Genomics Center. So it's a completely different study organism. I went from seals down to bacteria. It's definitely a big shift. But the genetics component of it was the same. So with my gray seal dietary work, there was a whole genetics component that I'll talk about later with the monk seals. So that's kind of what brought me here to Ramonis's lab, which was wonderful. I spent a little bit over a year at Bigelow, and then my full-time position there ended, and I still am on as a kind of contractual research technician. And about 6 months after that, I got a phone call completely out of the blue from National Geographic Channel, which was quite surprising because I had never put myself in any sort of lottery to do anything like this. And they said, you know, hey Dash, we have this program. It's a competition where we take 2 of our past young explorers, and we want them to compete for a second funding opportunity. And I was like, compete? Okay, tell me more. And they said, well, basically you propose a project, and then we'll pick 2 of you. You guys will make, you know, videos and TV commercials, and we're going to promote you. And by promoting you, you're in turn going to promote Expedition Week on TV. So basically the gist of it was, here's Dash, she's a young explorer. Vote for her, tune into Expedition Week, and find out who wins this competition. So they were using this competition to promote, you know, this special week on TV. And they said, do you want to put in a proposal and be considered? And we said, yeah, absolutely, I would love to do that. So then I went back to my grad school bucket of papers full of literature, trying to think, what the heck am I going to do for this? It needs to be a really exciting project. This is a whole social media and TV thing, and I really want to make sure that I'm having something people will feel passionate about, want to vote for, and that I feel passionate about, so that I can portray that and try and get the funding. So I stumbled across this paper from the very beginning of grad school when I was trying to decide what to do on the monk seals. And they are critically endangered, they're in decline, and I thought, what would be better than to get to go back to that original seal species that I was really interested in. So I put in a proposal to National Geographic where I proposed studying the diet of the Hawaiian monk seals to help protect the species. They decided to go with me and this one other fellow, Trevor, and literally 5 days later, I was involved in a nationwide full-on competition. And it was a little bit of it was definitely a bit of a shell shock from a phone call to a film crew within about 2 weeks. So they came to our house. They did 3 days of filming. We went down to UNE and filmed with the seals. We came to Bigelow and filmed there. We went to the beach. It was incredible. They did about 5 TV commercials that were all 30-second spots that were driving people to the website. And on the website, they had a whole page with lots of informational videos about me and my work and, you know, where I live. And that's all up still if anyone's interested. But I thought maybe you'd want to see one of the commercials. So I'm going to play this video. Sounds like the sound's on. It's not embarrassing.
Meet Dash.
I'm a National Geographic Young Explorer.
She loves monk seals so much she wants to study them and how they live to protect this endangered species. You can help her get her expedition granted. Go to dashgeotv.com/expedition and vote.
Vote for me and you can win a trip to the Galapagos. Wouldn't that be cool? Thank you. So yeah, we had a lot of fun with that, and let's just say that trip to the Galapagos was valued at 3 times my grant, but you know. That's okay. That's okay. So then the whole deal was National Geographic was like, okay, if you're in this, you got to be in this, and we need you to do as much social media, marketing, and promoting of yourself as possible. And I said, all right, I can do that. So I built a website. I built a Facebook page and a Twitter page. I went to Mystic Aquarium and set up a booth and got people to vote for me and handed out informational brochures. I made my Bigelow colleagues hold signs so I could take funny pictures with them. And then I drove everyone to this website day after day after day after day, and I'm sure lots of you out there can attest to the fact that I was slightly obnoxious because each individual could vote every single day, and I couldn't stand the thought of a missed opportunity. So lots and lots of voting. And then I ended up taking the grand prize, which was very exciting. So it was worth it. So it was great. We ended up Team Monk ended up being a really, really neat experience, and as I said to my husband, the one and only campaign I will ever have in my entire life. So glad to have that over with. But I ended up here, so that was not such a bad situation. So this is the island of Molokai. And how I ended up on Molokai was basically my only grant requirement for this grant was to bring back photos and video, which was an incredible, opportunity. I mean, usually there's reports and budgets and all sorts of stuff, and all I had to do was bring back pictures, so that was pretty cool. But the thing that's interesting about my project is that all the samples I needed were already archived in a freezer in Hawaii. So I thought, well, probably National Geographic doesn't want photos of the inside of the lab in freezer space. So I contacted NOAA. So the NOAA Pacific Island Fisheries Science Center Hawaiian Monk Seal Research Program is actually who I am working with on this project. So I contacted them and said, I really think I would like to get out in the field. I really want to see the seals, interact with the seals, and kind of fulfill this grant requirement for National Geographic. So we ended up on the island of Molokai, where I did a week tagging trip. This is kind of typical Molokai scene. It's a very desert island. It's very flat and desert-like. This is a hike we would do every day. It was an 8-mile loop. So we walked 8 miles each day looking for seals, and would stop at these rocky outcrop and then these little beaches and look for seals. So we spent a lot of time walking and watching. And then when we were lucky, we'd spot a couple. So these are a few guys that are hanging out, I'm trying to hide myself behind the bushes here. You can see, we don't want to disturb them because they're wide awake, so they would definitely see us if we moved too quickly. And then we try and ID the seal. This research program is unbelievable because they have such a large network of volunteers, and NOAA has been working with this species for so long because it's been endangered for such a long time, that a lot of these animals have flipper tags on them and they're already identified. And then a lot of the volunteers, if these animals aren't tagged, recognize them. So we actually have the ability to look at an individual seal and know who they are, how old they are, when they last molted, all this great information. So we spent a lot of time actually trying to figure out which seal we were looking at. And once we would select a seal, we would tag them. So it was this big kind of, exercise to try and figure out how exactly we were going to approach this animal, and there was a lot of stalking involved and a lot of crouching and a lot of whispering, and then we would basically, manage to capture the seal, and they would get a sedative, so that it was a calm process for both us and the seals. We were trying to get the tag on, and so you can see this fellow's holding a cell phone tag, is actually what they are, getting ready with some 5-minute epoxy here to tag the seal. And those cell phone tags tell us data on where they're swimming, how deep they're diving, all that good stuff. So this animal is getting tagged, and you can see here actually is the flipper tag that's on those animals with the unique ID. So we also at the same time collected biological samples, so no opportunity is a missed opportunity. We made sure to get blubber and blood, and I think maybe that's pretty much it actually, samples, but lots of good stuff out of that. And then we kind of reverse the sedative and send that little seal on its way, and it was just no worse for the wear. And then I looked for seal poo because I study seal diet, and you study seal diet by looking for seal scat, and that's super glamorous, of course, and everyone likes to make fun of me a lot for being a poopologist or a scatologist or loving poop, but there's a wealth of information in seal scat, so I would then look for seal scat. He'd kill me if I put this picture up here, but that is Alex Karamanlidis, and he is a Greek researcher with the Mediterranean Monk Seal program, and I'll talk a little bit more about all these seals in a minute. And he was wondering what Hawaiian monk seals smelled like. So that was an old seal call-out, and that's him with his nose to the ground because he knows what Mediterranean monk seals smell like, and he really wanted to know what Hawaiian monk seals smell like. That is Alex. So then, after my week on Molokai, we went to this island, which is called Coconut Island. And Coconut Island is right off the coast of Oahu. Literally, I'm standing on the shore here, so it's just a stone's throw away. And this is where the Hawaii Institute of Marine Biology, through the University of Hawaii at Manoa, has their marine labs. So it was here that I got to do my lab work. And you can tell I'm really tired at this point after that is National Geographic and the grant and getting in the field. So now I'm going to kind of go into monk seals, why we care about them, and what I'm trying to do. So monk seals have a lifespan of about 25 to 30 years. Males get to be about 7 feet and 400 pounds, and females are 7.5 feet and 600 pounds. So they're very big animals. They don't always look so big in these photos, but they definitely are very large animals. They're less gregarious than most other seal species, so there's not a lot of large congregations of them, and it's actually how they got their name, the monk seal, because they're quite solitary. They eat fish, eels, cephalopod, crustaceans. It's a pretty standard seal diet. And most of their feeding is around 75 to 90 meters, but they can dive up to depths of 500, which is incredibly impressive. And they actually used to think that monk seals were feeding primarily on shallow coral reef ecosystems. And they've done some critter cam work with National Geographic, and they're actually finding now that they spend quite a bit of time in more deep sea environments, atolls, sandy slopes, the likes of that. So they are actually having kind of a big shift on their view of monk seal foraging because of critter cam. So this is a brand new pup. Pups are born between February and July with a peak during April and May, so they don't actually have a large pupping season like we do in New England. In New England, all the seals come together, they have one big breeding congregation, they breed once a year, they mate, and they disperse. These guys are kind of a more year-round breeding cycle. They nurse their pups for 5 or 6 weeks, during which time the mother doesn't eat at all, so they might splash around in the water to cool off. Seals in Hawaii have to enter the water to get cool because they get too warm on land, whereas if you can imagine our seals here have to get out of the water to get warm on land. You know what that's like, right? So it's the same thing with the seals in Hawaii. And so they will splash around in the water a little bit, but the moms do not go out to forage, and they can lose up to 200 pounds, I think, during this time, which is pretty unbelievable. They kind of splash around with their pups and help teach them how to swim, but they don't ever teach them how to forage, so they wean the pups and then they're off on their own. So the pups have to make it on their own. Like I said, females can give birth on beaches with adjoining shallow waters, and that helps cool off and provide protection from sharks. And they first reproduce around 5 to 10 years old. Okay, so population as a whole, the monk seal are listed as critically endangered by the ICUN. There's only about 1,100 animals left, and the population is decreasing at 4% every year. So if you do the math, they're not going to be around much longer if something's not done at this point. A couple decades at most. They're the most endangered marine mammal in the United States and one of the most in the world. Second to the Mediterranean monk seal, which only has about 600 animals left, which is the only other seal species in the same genus, Monachus, as the Hawaiian monk seal. And there was a third species, which was the Caribbean monk seal, but they're already extinct. So this genus is, for some reason, just not really able to survive the pressures humans are putting on this species. Pups currently have less than 1 in 5 chance of surviving to adulthood, which is pretty grim. This is due to shark predation. So tiger sharks and Galapagos sharks prey on monk seal pups. Marine debris entanglement pollution is always a really big deal, and ghost fishing nets and all those plastic rings we read about, that's a real issue for these guys. Starvation, disease, and then male violence. So there's a lot of subadult males right now that are exhibiting, they're not necessarily breeding yet, but they are exhibiting a lot of aggression during breeding season, and a lot of pups get trampled or attacked, which is really, a hard thing. But the biggest thing here that they're actually finding is the cause of pup death is starvation. Okay, so historically, monk seals were found in the entire Hawaiian Island range. So this is the range we kind of typically think of as, the Hawaiian Islands, and then this is the northwest Hawaiian Islands. They're estimated to have been in Hawaii for 10 million years. The species has been around for 15 million years, and in the Hawaiian Islands for 10. They're the only marine mammal found exclusively in the U.S. So any other marine mammal that we see here in the United States also shares waters of other countries, but the monk seal is the only one who we are solely responsible for the conservation of. It's thought that that population was probably a few thousand to 15,000. That's not a ton for a seal species to begin with, but that doesn't mean that where they are right now is not still a critical point. They were quickly extricated from the main Hawaiian Islands when the first Hawaiians arrived, and I'm thinking that's about 800 years ago, so not that long ago. And the population as a whole, even in the Northwest Hawaiian Islands, was hunted to near extinction around the same time frame that was happening here, because of guano hunters, bird hunters, and people out to get the seals. Over the last couple hundred years, there's been some very rare sightings of monk seals back in this main Hawaiian Island chain. But recently, this new thing has happened where they're actually firmly establishing themselves. So there's now about, a confirmed about 200 animals living in that main Hawaiian Island chain and about 900 in the rest. And we have another really interesting thing happening with that, and that is that the main Hawaiian Islands are the only place where the population is actually increasing. So that's super interesting. Since the majority of pup death in the Northwest Hawaiian Islands is due to starvation, the hypothesis is that the pups are surviving in the main Hawaiian Islands because of a food source. Even though in those Northwest Hawaiian Islands, it's been a marine park for years, there's no fishing, there's barely any humans, I mean a couple hundred in the whole Hawaiian Island chain. So even though the main Hawaiian Islands have all the people and all the industry and all the fishing, there's still something going on there that's allowing these pups to survive better. So the goal of our study is to study the main Hawaiian Island monk seal diet to try and help save the species. Okay. Alright, so how do we study seal diet? I'm going to go through this really quickly because I feel like I'm already getting low on time. So conventionally, what we do is we collect seal scat, and then we take that seal scat back to the lab and we sieve it out and we look for fish hard parts. And hard parts for fish are otoliths, and then vertebrae, islands, cephalopod beaks, any kind of hard structure. And the fish otolith is an ear bone of the fish. And each fish species develops an ear bone that has a unique species identification. So you can tell two different fish species apart simply based on their ear bones. And so that's what we do. We sieve out the scat and we look for all these fish ear bones, and then we try and identify them to species level. There's a lot of controversies with this though. And the biggest one is that those ear bones can erode to the point where you actually can't recognize them any longer, so you can't tell what species it is anymore. Some of those hard parts disintegrate altogether, so you never even find them. Lack of hard part ingestion, there's this whole theory where seals belly bite, and it's when they capture a large fish, and they actually just rip out the calorically rich gut region of the fish and never even eat the head or the backbone or anything. So you never even see those hard parts. And it's a short time frame. It's only what they ate in the last couple days. Okay, there's other methods for determining diet, stable isotope analysis and fatty acid analysis, and then fecal DNA. All of these things have biases as well. Stable isotope only gives you trophic level analysis, so you can only kind of see where in the ecosystem the seals are foraging, not what specifically they're foraging on. Fatty acid analysis is this whole theory that each fish has a unique fatty acid signature, and as the seal eats it and incorporates those fatty acids into its tissues, that signature remains, so that if we then look at the fatty acid in the seal tissue, we can see what fish they're eating. That is a really great idea in theory, but in practice that the fatty acid profiles do not perfectly integrate, and there is a lot of changing and there's a lot of modeling involved, and it's a lot more complicated than it might seem, so there's a lot of biases with that. And the fecal DNA, like looking for hard parts, has a pretty short time frame. So our best bet with studying seal diet, especially with a really important species like this, is to use multiple methods. So what I'm doing with this project is using fecal DNA to complement fatty acid studies and hard part studies that have already been done. So I'm really lucky to get to come in and kind of help put some missing pieces in a puzzle. All right, so this is the puzzle we have. This is a fatty acid analysis of Hawaiian monk seals. The top two, I know you guys can't read that, it's really blurry, but it's okay, I'll explain it. The top two are Northwest Hawaiian Islands, the bottom one is main Hawaiian Islands. So if you see those main Hawaiian Island population of monk seals, there's that huge bar, right? I mean, it's unbelievable. It's over 30% of the diet, and that fish species is called flower snapper. So in theory, according to fatty acids, these Hawaiian monk seals in the main Hawaiian Island are eating 30% of their diet as just flower snapper. But what's really interesting about that is that the Northwest Hawaiian Islands did a hard part study. So this is a different region, but it's still the same species. And they looked at 59 spews, seal vomit, which is probably even less appealing than seal scat. And they found 759 different fish and 35 different taxa, and not a single flower snapper in all of that, okay? And then in the main Hawaiian Islands, they just finished, and I don't have the data with me because they've literally just finished, it's not even published yet, scat analysis, hard part analysis in the main Hawaiian Islands, so this same population, and they haven't found any flower snapper in hard part analysis. So there's this big discrepancy, and how do we protect a seal species if the only way to protect them is to protect what they're eating, but we don't really know what they're eating? So what I'm trying to do is to come in and use fecal DNA to try and really help answer this question. And on top of that, you know, maybe if this species was a species that no one cared about, no one was fishing for, it wouldn't really matter because there would be nothing we could do about it anyway. But this flower snapper happens to be one of the 7 most important commercially fished species in Hawaii. So it adds a whole other component to the puzzle, which means, you know, we obviously need the Native Hawaiians and the local Hawaiians to be able to sustain themselves through fishing. You know, I grew up in Maine, I know how important fisheries are to our economy, but we also need to figure out how to coexist with the seals, so we really need a clear picture in order to do that. So we use molecular ecology. And basically molecular ecology is just using DNA to ask an ecological question. So my question is, I want to see if Hawaiian monk seals are eating flower snapper by looking for flower snapper DNA in the monk seal scat. And if I can find flower snapper DNA in monk seal scat, then there's only 2 things it can be. It's there or it's not there. So it's presence, absence data. It's not going to give us any information on how much they're eating, or even really how often they're eating it, but it will tell me if it is a component of the diet or not. And since we have one study that says it's 30% and one study that says it's 0, that can be some really helpful intermediate information. How we do this is we use mitochondrial DNA for fecal studies. Mitochondrial DNA is great for fecal studies because it's present in multiple copies in each cell. So you don't just have nuclear DNA, you just have 1 copy in every cell. So if you get multiple copies in each cell, with all that degradation that's happening during digestion, you have a better chance of finding DNA. And there's also extra membranes around the mitochondria that help prevent it from degradation. Specifically, what I'm looking for is the CO1 gene. This gene is used by a lot of scientists due to its mutation rate. So this gene mutates at a fairly quick rate, which means that there's a lot of differences in this gene region between species, but it's actually quite well conserved within a species. So it's a great gene region to use if you're trying to ask the question, what species is this? I have DNA, what DNA do I have? And on top of that, there's a database called BOLD, and 620,000 specimens from 58,000 different animal species have already sequenced this gene region, which happens to be very helpful when you want to use that for a research project. Now, of course, the flower snapper has not been sequenced, of course. So, quickly run through how I'm going to do this process. I sifted through 3,000 fish samples that were frozen and macerated and frozen in Hawaii, and I managed to find the flower snapper, which was a miracle, and basically took a little fin clip from the fish. Okay? So now I have this little piece of tissue, and I'm going to extract the DNA from that tissue. Really simple process. You digest that tissue, you lyse all the cells, you expose all that DNA, and then you put it through a little tube with a filter, and the DNA binds to the filter. Then do a couple washes to wash away all the impurities, and then you do an elution step, which is basically you take this buffer that then convinces the DNA to release from the filter and come into the tube, and now I have a tube of liquid full of DNA. Okay? I then can take that flower snapper DNA and I can put it in a reaction cocktail in this plate. Basically, in that tube, I only have the DNA that has come out of that tissue. That is not enough DNA to actually work with and study, because I need to be able to visualize the DNA and sequence the DNA. So I need to make a lot more copies, and we do that through PCR. And just a quick, you know what? Maybe I'm going to skip this video on PCR, actually. Well, it's quick. This will help.
Chain reaction for PCR uses repeated cycles of heating and cooling to make many copies of a specific region of DNA.
So here's my flower snapper DNA.
First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the prime sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Okay, so the important thing there is these primers go into this reaction, and these primers are what allow us to isolate a certain gene region from that DNA and make many, many copies so we can study it. Okay, so now we get something like this. We get clear liquid in a tube again, and then we send that liquid off, and they sequence the DNA, and then you get a data file back that looks like this. So this is actually CO1 data on pink Siebold's and red snapper. I have not actually yet done this with my pink snapper. My flower snapper. Still trying to get up and running. But these are pre-published sequences on a database called GenBank, and GenBank is kind of like Bold, but GenBank is the world's largest database of DNA sequences for all different gene regions. And when most scientists will sequence something that's never been sequenced before, they deposit it into GenBank so that we can all access it for research. So now I have my CO1 gene sequence, and what I want to do now is make a flower snapper specific primer, so that when I put that primer in a PCR reaction, the only thing it will amplify is flower snapper. And the way you do that is when you look at this sequence, you look for regions with a lot of variability. So the stars are where the nucleotides match, the blanks are where they don't. So what I basically want to do is find places where I can make a primer that will match perfectly with every single flower snapper base pair, but mismatch with every other fish out there. Really easy, obviously, and then get the reaction just to amplify flower snapper. So that's really the goal of making species-specific primers to then be able to use in this study. So then once I get these primers, I blast them, which is just searching in GenBank to see if these primers will match with anything else. So this is a good theoretical search. Basically, I don't want the primers to match with anything else except what I've designed. So I search for similar sequences online, and then I do lots and lots and lots and lots of control testing. So I'm going to test these primers with lots of other fish. I brought home about 50 fish species from Hawaii to test these primers against to make sure that they're not going to amplify anything else in the seal diet. And then, of course, I test the primers with seal tissue because I don't want them to amplify the tissue of the monk seal either. And then this is what we would hope to see. So this is DNA that's been amplified. It's been stained so that we can visualize it, and it's run on an agarose gel, which is just a matrix that we run a current through and get the DNA to separate out on. And what we basically hope to see is when I put this DNA from a given seal scat in this PCR mixture with my flower snapper primers, I would want to see flower snapper amplified. Sorry, I should back up. That's tissue, not seal scat. So when I put flower snapper tissue in this reaction, I would want to see that it amplifies perfectly every time. And then what I would want to see down here is that these are the reactions where I added monk seal tissue, red snapper tissue, Siebold snapper tissue. I wouldn't want to see anything, meaning my primers will not amplify those other species, just the flower snapper. So that's the goal. And then the last thing I need to do with this DNA is, so I've now proved that in theory by blasting these primers, they only amplify flower snapper. I've proved in the lab controls they're only going to amplify flower snapper and nothing else. But the last thing I need to do is prove that once flower snapper DNA is ingested, digested, and come out on the other end of a monk seal, it's still recognizable as flower snapper DNA. So this is Maka, and Maka is who I'm going to be working with on feeding trials. So I will feed Maka flower snapper. I will hope to goodness that he defecates on the deck of his pool and not in the pool, and then I will analyze that DNA or that scat sample for flower snapper DNA. I'll show you a quick video of him because he's great, and he has heard an announcement on the intercom right now that in 5 minutes there will be a feeding at the monk seal exhibit. And so he's heard this automated voice over the loudspeaker, and this is why he's making a ruckus, because he knows it's coming.
Aloha and welcome to the Hawaiian Monk Seal habitat. During the feeding and training session, I'll be telling you a little bit more about this interesting marine mammal. I'd like to introduce you to our resident male. His name is Maka Onauna. That's Hawaiian for soft or gentle eyes. Maka arrived with us in 1984 from the French frigate Shoals as a gold pup. This is pretty unusual. Pups are normally nursed at least 4 to 6 weeks. Maka was found without his mother, and he had either been orphaned or abandoned. Due to his small size and young age, he most likely would have not survived out in the wild. He came here to the aquarium, and he is currently about 27 years old, 7.5 feet long, and he weighs about 350 pounds. What I'm doing with Maka is what we call the daily body exam. We're able to do these 2 to 3 times a day at each of our feeding sessions. We can check over his general health, looking for any cuts, scrapes, bumps, or anything out of the ordinary. These exams also serve as good practice for Maka. It keeps him used to being handled by humans and having his various body parts examined. You'll notice that Maka is touching his nose to a blue and white float. This is a training tool known as a target. There is no smell, sound, or taste associated to this. It is simply a trained behavior, and Maka has been trained to touch his nose to the end of this float. He knows that in doing so, he will receive fish as a reward.
So that's Maka, and when I go back, that bucket will be full of flower snapper and not what he's getting there, so which is probably something more delicious. But so then I now hopefully have developed flower-specific primers. I tested them against every tissue imaginable. I've proved that they can be that they work when fed to a captive seal. And then at that point, I just get to go with these environmental samples. I would extract the DNA from all the scats. There's 100 main Hawaiian Island scats that have already been collected and archived in Hawaii. And then I would do PCR for presence absence data for flower snapper on these scats. And then hopefully that'll get some really good information to help complement the fatty acid and the hard part study with the monk seals. After that, we actually want to try and do about 2 to 3 other fish species. And then we try and use that data to help protect the monk seal's diet and coexist with fisheries, both very important things. And NOAA, of course, would love me to analyze their 700 Northwest Hawaiian Island scats, but that's certainly down the road. I really just want to make sure I thank National Geographic, NOAA, University of Hawaii, and of course Bigelow for having me here today. So thank you so much. And questions? Basically, we tried really hard with wild animals not to do that because they get too accustomed to humans in that situation, and then they probably wouldn't be likely to survive on their own in the wild. But what they are trying to do right now is they're trying to fund a hospital through the center in California, which I will come up, Marine Mammal Center in California. And they want a rehabilitation facility. So they have nowhere currently to rehabilitate pups. that may have been abandoned or injured, or even adults that may be wounded. So that's what we do at the University of New England with harbor seals here. And Hawaii is trying desperately to raise money to have a facility like that, which could be really helpful. Yeah. Yes.
Is it possible that the ecology of the diets of monk seals in this area, they are in this area?
In this area? No, no. Yeah, great. Okay. In the back.
What do the monk seals in the northwest area of Hawaii eat? That's different from Hawaii.
Well, they eat similar species, but it's really just more the composition of the diet that we're interested in. So, you know, we know what seals eat because they're generalist predators in theory, so they eat lots of different things. And so we know that they're eating, you know, squid and fish and eels and all that, but we're really interested in kind of the different levels. And that's still debatable. The last hard part study in the Northwest Hawaiian Islands was from 1999. The fatty acid showed some differences, but that's really the question we're trying to get at. What is the major difference? Yeah.
Do you have any combination where you have the hard part and the fatty acid study out of the same animals so you can look at that variance and know the meaning of that?
Sure. So she was asking if we ever see fatty acids and hard parts with the same animal. The thing that's hard about
you know, so
sure. What's hard about that is that fatty acids we get when we do tagging because we take a blubber sample and we can get that data from tagging. So we always know what animal the fatty acid is coming from. Scats are actually found usually opportunistically on a beach, and there's usually no animal around. So tying a scat back to individual is much more difficult. One of the things I would love to do with fecal DNA, though, eventually, is to start getting into looking at individuals. So in theory, I should be able to take a blubber sample and genotype the individual, and then I should be able to apply that same theory to the scat and try and make those connections, which would be incredibly interesting. Time frame might still be off, but we could still see an individual's kind of diet over time. Yeah.
I want to ask a question about education and outreach as a scientist. Clearly it's important to your work, but how do you feel about social media? In particular with your experience with this project and the pros and cons, if you would.
Sure. So the pros and cons of social media in science. I never would have thought of doing something like this. I certainly never would have. I really applaud National Geographic actually for pulling social media into science because it's just something I never would have thought of. But the response I got from people was incredible. I got so many emails on, you know, you're inspiring and I love what you're doing, to this is really what I want to study and I've always wanted to do this too, to I had no idea that these seals even existed and were in such trouble. So I got lots and lots of positive response from it. I still don't know. I don't know. It's interesting, and I think as an educational outreach tool, it's certainly interesting. And you see now on Facebook tons and tons and tons of nonprofit organizations who are spreading the word and, you know, have a lot of followers. So it was great. It was great. But it was a lot of time and effort and work, and I don't know if really, you know, targeting a group of students in Hawaii in a classroom might be more effective than targeting individuals. While it takes the knowledge and really spreads it out, I don't know if it really is as effective as making changes on the ground. So I'm not sure though. Yeah.
I'm somewhat confused because I have seen documentaries where scientists are on the outer islands.
Okay.
And they're covered with plastic.
Yeah.
And the animals are eating plastic and they have emptied their stomachs and found plastic.
Yeah.
And that's why they're dying. They can't absorb the nutrients, plus they have the poison.
Sure.
And you were on Molokai, which is cleaner, there's less plastic.
Yeah.
And it may be just a difference in them not eating plastic. And it seems logical that you might test the scat for plastic. So
Yeah.
how do you incorporate all that?
Sure, that's really interesting. That's an interesting. Did everyone hear that? Yeah. I haven't gotten feedback from NOAA that is something that they really would like to look at, but it's certainly something that we could do. We could test scat for plastic. I actually just get a little subsample of each scat to put through DNA analysis. It's really the person doing that hard part analysis that would want to look for the plastic particles in the scat. So it's kind of a different part, but and I actually hadn't heard that that was a big cause of their starvation.
Oh,
so.
it's pretty much scientific. It's there. I mean.
Yeah, no, I definitely know plastic on those outer islands is a huge issue. Yeah, yeah, scat would be a great way to look at that, definitely. So, yes.
How I've done this is when you tackle this seal,
Yeah.
you get the blood sample. Could you get a sample from the stomach and from the intestine, both ways
Sure.
at the same time?
Sure.
This way you've got the scat.
Right.
Seal.
Right. Okay, so he was saying when we tackle the seals to tag them, could we also take a stomach sample and a scat sample at the same time? So I'll get a little graphic because it's a graphic topic, but they do fecal swabs with a loop, but that's not enough to actually get a scat sample per se. And the thing about these seals is we're trying to be as minimally invasive as possible because they're so endangered. We want to kind of get our equipment on them and get them on their way without causing too much stress. And so, putting a tube down their throat or an enema or something would be, I think, a much more traumatic experience. And I don't know, people might do that, but that's certainly not what Noah wants to do out there. So sometimes the seal gets freaked out enough that you get a scat sample though. That's good. Yeah, and we're really excited about that because we know where it came from, so that's exciting. Anything else? Oh, yeah.
Do you know how stable the flower snapper population is?
I don't actually. That would be definitely interesting. I'm not quite sure because I've just started kind of working with this. I don't even know what the annual landings are really. But I do know that there's not a huge commercial fishery in Hawaii, but there's also very little regulation because of that. So I don't think there's a ton of monitoring. So I don't know if there's really a ton of information available. And it's kind of more like, if it's important, we'd like to make sure it's around for a long time to come. So.
What is the predominant way that the monk seals pass their feces, on land or by sea?
Oh, definitely by sea. Definitely, yeah. Most defecation happens at sea. That's why over 5 years in the main Hawaiian Islands they have 100 scat samples, which if you think about a population of only 200 animals, that's pretty good, but it's still not a lot. And I spent so many hours looking for seal poop in Maine, it was unbelievable. And actually found that harbor seals go to the bathroom on land way more than gray seals, of course, and I couldn't believe it. But yeah, no, at sea is definitely more common. Yeah. Sorry?
There was you said something about amplifying primers in the flower snapper.
Okay, yeah.
What do you mean by amplifying? What does that term mean?
So the primers I create are that little flower-specific molecule that when I put it in a whole reaction full of DNA will only amplify flower snapper DNA. And by amplify, I mean make millions of copies so that I can then study it and visualize it on those gels that I showed you a picture of. Does that make sense?